Good afternoon, everybody, and welcome to the webinar. We are in a brand new webinar series today discussing clinical trial education. And we are so excited to kick this series off with a really important topic, and that is the importance of clinical trials, why they matter, and why anybody should care about clinical trials. Before we get started, I wanted to give you a little bit of information about Mended Hearts. Our mission is to inspire hope and improve the quality of life for heart patients and their families through ongoing peer-to-peer support, education, and advocacy. You can find us online at any of the social media accounts or at our website, mendedhearts.org or mendedlittlehearts.org. Before we begin, just a couple of notes. All of the attendees are in listen-only mode. If you cannot hear, hopefully you can see the slides, and it's asking you to check the audio button on your personal computer to make sure your sound is on. That's typically the problem that we see, and that can help you out really quick. Please type your questions into the Q&A box at any time during the presentation, and the moderator will read your questions out during the Q&A period. Please note that the presenter will not be able to answer specific questions about you or your loved one, as they are not the treating physician. And finally, the PDF version of the slides, along with the recording of this presentation, will be available on the Mended Hearts website following the event. One more note, there is a PDF handout in the chat right now. There is a link where you can go to the website and download the handout, and it is a discussion guide about clinical trials. So without further ado, I'd like to introduce to you Dr. Charles Davidson. He's a Vice Chairman of Clinical Affairs at the Department of Medicine Clinical Chief, Cardiology Medical Director at Bloom Cardiovascular Institute, Professor of Medicine. So Dr. Davidson, I'd like to turn this over to you, and thank you so much for being here with us today.
Thank you very much. It's actually my pleasure to be able to speak with you about, I think, a very important topic that I think is really maybe underappreciated and maybe not even well understood for people that are not baked in cardiovascular medicine. And that is, why do we do clinical trials? And before I get into the data to really tell you what we do in clinical trials, I think it's important to understand what is a clinical trial. So a clinical trial is a research study that's performed typically to prove the safety and effectiveness of a therapy compared to standard therapies. So let's say medicine is a standard therapy, and we're trying to show whether a certain treatment, let's say a valve or whatever, which is what we'll focus on today, is better than medicine. The FDA will say, before we can approve that device, we want a trial. And typically these trials will be half the patients or 2/3 of the patients will receive the device, and a third or a half of the patients will receive standard therapy, and then we compare their outcomes. And so that's what I'm going to kind of go through today, is clinical trials that have been done over the last few years to prove the benefits of catheter-based therapies or non-surgical therapies for the treatment of patients with valvular heart disease. So, kind of move to the next slide. I'm having trouble advancing. Okay, so aortic stenosis. So this is the main valve that leads out of the heart, or the aortic valve. It connects the heart to the rest of the body. And so when patients develop aortic stenosis, it is a buildup of calcium within the valve. And many people ask, well, is this something that was dietary or something I did wrong, or my cholesterol or whatever? No relationship to this type of calcium. It's different than heart artery disease, where we know cholesterol-lowering drugs and other lifestyle modifications can affect this. Not so with valvular heart disease. There are people that are born with abnormal valves, and that's a bicuspid valve, meaning they only have two cusps, 1 and 2. Those people are more prone to developing calcification over time, particularly in their earlier years. As you can see here, often this presents in people's in their 30s and 40s and even 50-year-olds. For tricuspid disease, which is a normal valve, you can see it has 1, 2, and 3 cusps. There's calcium that's built up over time. And this is something that happens usually later in life. So we see patients in their 70s and 80s, 90s, and even I've had 100-year-olds that have presented with this problem. And so let's talk a little bit about standard therapies here. Standard therapies were medications, and those were used for years. Now I'm going to show you a number of these kind of graphs. And so what this graph looks at, it looks at people that developed aortic stenosis and then follow them over a period of time. And then what happens is they do just fine with aortic stenosis, but once they develop symptoms, and we look at how many patients survive, we find that within 2 years, people developing symptoms of heart failure, which is shortness of breath, difficulty breathing with activity, within 2 years, medical therapy, almost half the patients will die. And same thing if they develop angina or if they develop passing out episodes. So we knew, and this study was done years ago, that people with medical therapy and aortic stenosis had to have the valve replaced. Medications don't work. It's like Drano, when your drain is completely clogged, you need to unclog the drain. The Drano is no longer going to work for that patient. So what did we find? And before I get into that, I want to show you what occurred really in the last 10 years. It's been amazing. I've been involved with this since we started back 15 years ago. But it used to be the only treatment for these patients was what we call surgical aortic valve replacement. And you can see how many of these were being done in the United States. 20 to 30,000 of these were being done a year. And then suddenly transcatheter valves came in, and really they came in around 2005, 2026, 06. And what happened was over time, and this is when it finally got approved in the US, it has now replaced surgery as the #1 treatment for people with aortic stenosis. And that happened in 2015. And really this line has been a straight up line even past there. So just looking at what happened at Northwestern. Now we're a large referral center. We see a lot of patients with aortic stenosis, but the same thing happened here. We developed this, we started, as I said, around 2008, we were doing 5 a year, 21 a year, and this only goes out to 2017. Now we're up to over 300 of these being done a year. And what has happened though, when you look at the slide, is the amount of surgery that's been done. Used to be 93% of the patients came in with aortic stenosis got surgery. Now it's less than 25%. Our most recent data is somewhere around 20%. So people might ask, well, gee, surgery worked well for a long time. Why did that actually occur? And so there were multiple things that happened. One is the technology. We're now in our 4th generation of this technology. It was actually a generation even after this. The first ones were a little large. You can see they had catheters that were about the size of a small garden hose, really, that we needed to be able to get in through someone's femoral artery. Well, you can imagine a lot of people didn't have that type of anatomy. So we had to do them alternative methods, even through the tip of the heart we were putting them in. But over time, we got smaller and smaller catheters. The other thing that happened was there was a skirt that was put around this so that when this valve went in, there was no more leaking around it. Now, many of you have never seen a valve, so I'm going to just explain to you what are the components of a Tavr valve, a transcatheter aortic valve. And that this one has cow pericardium, so the lining of a cow heart is sewn into the shape of a valve, and that is mounted on what is a cobalt chromium, used to be stainless steel, so a metal frame. So many of you are familiar with what a stent looks like. Essentially, this is a stented valve that is put in over a balloon, and I'll show you exactly how this happens. And then this skirt is placed around here to make sure there's no leaking around the valve. So this was a terrific innovation when we started these trials, quite a long time ago. So here I'm going to just show you what is a Tavr. So I'm going to just pause this for a second so I can give you some of the landmarks, because I know many of you have never seen what I'm going to show you here. I want to give you time to look at it. So what we've done now is we've put in a tube from the leg artery, can be the right or left leg artery. And about 90% of people these days have anatomy that can accommodate this kind of tube. For those of you who may have had an angiogram, this is a tube that's about 2.5 times a normal angiogram tube. This comes up from the leg, it goes across the aortic valve, and then we place a wire down into the apex to the tip of the heart. And then this delivery system is then placed across the native aortic valve. We have a little catheter here so we can take some pictures. And then what we're going to do is we're going to inflate this with a balloon. So there's a balloon underneath that same stented valve that I showed you. So the stented valve has been closed down, clamped down, so it's very tightly adherent to the balloon. So we can navigate across the valve, navigate through the arteries, and now we're going to then inflate this balloon. So what we do is we pace the heart at a fast rate in order to lower the blood pressure so that we can precisely place this. And you'll see here in a second, the heart's going to be going fast in a second. And then you'll see us actually inflate the balloon here. This is the balloon inflating on both sides. You can see the valve now going up. It's being placed in there, and it's anchored by the calcium that I showed you in the native valve. So all these valves are calcified, and that calcium is sufficient to anchor this valve. And honestly, when we started with this, whatever it was, 14, 15 years ago, I was very skeptical that that was going to be a durable and good solution. But having done now thousands of these, it works, and it works very well. And it's a procedure that can be done, usually takes me in about 30 to 45 minutes. We don't use general anesthesia anymore. Patients are just under moderate sedation, kind of like if you've had a colonoscopy. And many patients are able to go home the next day. So this was a terrific innovation in the treatment of therapy for valvular heart disease. And then after we've placed the valve, you can see right here, this is how it looks. It's being anchored, as I said, by the calcium in the native valve. And then this is us just doing an aortogram, injecting some contrast dye so that we can see if there's any leaking. And in this case, there was no leaking, and that, an excellent result. We put a couple stitches in the artery and the tubes come out. So these are curves that we call survival curves, and they're kind of the hallmark of clinical trials. And once again, I'm not looking to make you an expert on them, but these are what we use and what the FDA uses, and what you should want to understand is how well do people do with one treatment A versus treatment B. So in this group, when we started with extreme risk patients. So these were 80, 90, 100-year-old patients that had no surgical options. They were just too sick to have surgery. So half the patients got the standard medical therapy, which, as I said, we knew historically didn't work. But the idea was, does this valve work at all? Really, can we take the people that are not surgical and actually treat them with this transcatheter valve, and can we do anything? And so we look at what we call all-cause mortality, basically who was alive and who died. And then we looked at them at 12 months, 24 months, 36 months, 48, and then the study was carried out to 60 months, essentially 5 years. The first thing to notice is within 12 months, and this was really the part that drove this device to be approved, is that 50%, as I told you, 50% of patients that got medical therapy did not survive. 30% of the group that got the catheter-based valve died, but 70% survived. So we had an improvement of survival of 20% in the group that received the catheter-based valve. That's a huge treatment effect when you think about it. 1 out of 5 patients were able to get better and survive by this. And the other piece that was very encouraging is that we followed these people out for many years, and we still maintain that almost 20% survival benefit. Now you might say, well, that's still a large number of patients that are dying, but remember, these are patients that really were very sick. They had bad kidney disease, they had coronary artery disease, they had bad heart muscles. These are patients that were very elderly. So they really had limited or no options. And we had brand new technology. Nobody had ever done these procedures before. The technology was very rudimentary, I would say, at the time, in retrospect. But at the time, it was saving lives, and saving lives in a big way. And that led to the initial approval of the transcatheter aortic valve replacement. So what I was showing you really comes forward as you start seeing, as the technology improves, and the operators improve, we got better. So when we were looking at essentially what the event rates, what was mortality at 30 days in the first trials, it was 6%. Again, sick patients, new technology, new operators. As we move forward now, and we get into this lower risk population and better technology, 30-day death rates now are actually less than 1%. I'll show you some data. So once again, everything kind of moving in the direction. And this is what we found all through clinical trials. These are multiple centers all throughout the US performing the trials in the exact same way, randomizing the patients and getting these results. So the best trials are what we call prospective randomized. That meaning that we're looking at the data forward. We're not looking at patients who have been treated already, but how they are treated moving forward. And they're randomized and they're in multiple centers. Because then you can say, is this reproducible from center to center, at least in the 40 best centers that were involved in this study. This was just another study looking at it. As things got better, we went on to now treat low-risk patients. So someone comes in 65 years old, no other heart problems at all, but wants to know whether they can get a Tavr valve or a surgical valve. And so we had to do some studies, same thing, clinical trial, randomized low-risk patients to surgery versus Tavr. What did we find in that? And this was called the Partner 3 trial. The risk of death or disabling stroke in the Tavr group was 1% at 12 months versus 3% in the surgical group. This actually astounded me. I always thought, well, if we could prove we were even as good at surgery and getting results, because surgeons were getting great results and still do. But if we could actually prove we were as good, I would have been happy. But this actually showed in this study that there was a benefit to not having open surgical approach. The other thing that people ask is, what about recovery? And this is, once again, clinical trial, same trial. We collected data on how do people feel at 30 days and 1 year. And Tavr here in the blue, yellow is surgery. As you would expect, if you don't have an open chest procedure, people actually got better quicker. At 1 year, it was all about the same, but within 30 days, people felt better. NYHA class is a way we determine how people feel as far as shortness of breath. They were able to walk further at 6 minutes at 30 days, although these differences were the same at 1 year. And their overall feeling of wellness, this is a questionnaire, also was better. So when you ask people via questionnaire, you ask them to walk 6 minutes, or you ask them how their shortness of breath was, all better in the short term, but the same in the long term. And I think that's when we're counseling patients, this is where we use the data from the trials to be able to give you information to say, okay, we can give you a quicker recovery, but overall the outcomes look the same. Now, I think the one thing we have to be careful with is that when we came across these transcatheter valves, remember the high risk were quite a while ago, but now as we're doing a low risk population, what are we going to find when we're out 12, 15 years? Because we know surgical valves, usually the biological ones, last about 15 years. Obviously, there's people last 20-something years, there's some that last less. But if we want to shoot for that 15-year time frame, it's going to take us time to show that durability is the same. But it is the same material that the valve is made of as far as the valvular material. They're made by the same company, and the difference is it's on a frame compared to being sewn in around the old valve. And the old valve still stays in place with this one. For the surgery, the old valve is taken out. So we need more time to know the durability. And I think this is where talking to patients and saying, look, this is what we know from the clinical trials, but here's what we don't know. And if they're very young, I think this is where discussion and a shared decision-making is very important. Obviously, everybody would rather avoid surgery if they could, but there are some unknowns, particularly in the younger populations. So we really indicate that the relative value of Tavr compared with surgery is independent of surgical risk. In other words, we started with extreme, we went to high, we went to intermediate risk, but now low risk. So now the guidelines for treatment of valve disease have actually changed in the last 6 months, saying that basically it doesn't matter what your risk category is, either one is reasonable for treatment of patients with symptomatic severe aortic stenosis. And I think the important point here is it's really shared decision-making process, and you have to understand the patient preference, which we talked about, understanding the knowledge gap, as I said, in younger patients, and considering the clinical and anatomical features. I won't get into too much detail with that, but all of these things were all occurred really within a 15-year time frame. And I'd like to show this slide as a bit of a joke, because Mick Jagger, several months after having his Tavr, was out dancing on the stage again. And as you know, his chest has never had a sternotomy done to it. But he's one of those people that really is a great ambassador, if you will, for this technology, on how he got back to work in no time and actually is still outperforming. So I'm just going to talk about a few other things that we're working on. You know, what else can we do with valvular heart disease? Aortic stenosis has been the big one. What we're also able to do now, this is a patient that had a previous mitral valve placed, a bioprosthetic mitral valve, so biological mitral valve, was placed in about 10 years ago, and that valve began to fail. The valve began to have blockage in it. And so now we have the same Tavr technology that we can cross over from the right side to the left side of the heart, and then we can place that same sapien valve into the existing biological valve using the frame rather than the calcium of the valve as an anchor. And this is a nice result here. I'll show you the pictures in a second, but this is a very nice procedure. It's done from the vein, crossing the right side of the heart to the left side, coming in across the valve. Again, we put a wire out. Everything's always done with a wire. And then we have the inflation of the valve and a treatment of a new valve without having a second operation. Other things we're doing in the mitral space, this is a mitral clip. So this is an interesting technology. What happens with people that have leaking mitral valves, this is the valve that connects the top part of the heart to the bottom part of the heart on the left side, can develop a situation where the leaflets of the valve are not coming together properly. So they, instead of coming together and closing, they come together with a gap between them. And so with that gap, we can use this almost as a clip, like a staple, if you will, to grab the leaflets. And this little white thing here shows us grabbing these leaflets, pulling them together, and then cinching up these leaflets. And maybe it'd be easier for you to see it on this bottom panel, where you can see that this thing has been placed in. Again, we come from the right side of the heart into the left atrium, across the mitral valve, into the left ventricle. This clip is then placed under echo guidance. And then once we have it in place, we release the clip, and it keeps these leaflets tucked together where they were not working properly before. This has been approved for people that are generally high risk for surgery. And right now we're undergoing a trial for intermediate risk. So very similar type of trials I explained to you in the aortic space we're doing now in the mitral space to try to move down that risk profile. Most of these trials start out with people that are very high risk, because we know that surgical options and medical options are not necessarily optimal. And therefore, you know, it's a bit of a lower bar to prove that they're better, but it is a place to offer patients treatments early on, because these may be years before they're approved. And so having an option for patients prior to approval, I think is one of the clear benefits of clinical trials. And what we've done in partnership with the FDA has been phenomenal, because the United States actually used to be a little slow at getting trials even started here. And we were often with Tavr, for example, we were 3 to 4 years behind Europe in getting those devices to be able to use for patients. And the FDA really in the last several years has taken a new approach to this, wanting to help patients in the US early on, with obviously full disclosure to the patients of what we know, what we don't know. And I think that's one of the most important things is, if you agree to a trial, you know, how many have been done? What's the risk of it? How many have you done? What are the safety issues you've seen? How durable is it? You know, these are all questions we may or may not have really full answers for, but need to be part of a discussion. So using that mitral clip, again, I'm going to show you one of these survival curves. These were people that had leaking valves as a result of a weak heart. So they may have had a heart attack, they may have had a myopathy, they may have had longstanding atrial fibrillation that weakened their heart. But we know that surgery never worked well there. And we know that medications, what we call guideline-directed medical therapy, actually worked pretty well. Once again, clinical trial, randomized, multiple centers, patients follow up for multiple years, all-cause mortality. What we saw in this trial at 2 years, that if people received medical therapy, GDMT, guideline-directed medical therapy, half those patients died, but those that received medical therapy with a clip, only 30% died. So once again, almost about a 17% improvement in overall death, which means that the number needed to treat, if you treat 6 patients with this device, you'll save 1 life. And that, you know, to us is a phenomenal treatment effect. They only have to treat 6 patients to save a life. And this is another one totally unexpected. I remember when we did this study, and I remember when the data came out, there was a standing ovation at the conference of thousands of people, because I think nobody expected to really see this kind of separation of the survival curves. And what was interesting was, if you follow them in the first 6 months, their actual survivability is about the same. It got better at 2 years, in 1 year, 2 years better, and now there is 3-year data that shows that these curves continue to widen. So this is a therapy, once again, through clinical trials, we proved was better. What was allowed in this trial, which I think many of the trials are allowing now, is if you were in this group that got randomized to guideline therapy, once they reached this 2-year endpoint, assuming they were still alive, of course, they were able to cross over to the clip alone, because there was no, it took another year for the FDA to actually approve this technology. So the only way people could get this, again, was in clinical trials, those that had agreed in the beginning, and then there was a registry that occurred afterwards. So there were ways of getting this device prior to commercial approval into many centers. Usually there's a limited number, as I said, usually 40 or so. I'm going to spend a few more minutes here, and then I want to leave it open for questions. What's really exciting now is we've also moved into valve replacements for mitral and tricuspids. We talked about the mitral valve. Many of the patients, we can pull that clip in there, but some of the patients have such a large gap between their valve leaflets that a clip's not going to work. And the same thing happens on the tricuspid side, which is the valve that connects the top and bottom of the heart on the right side of the heart, where blood goes out to the lung. Once again, often those leaflets have been so far stretched out that we can't actually get them to come together with a clip, and in which case a valve replacement. So there are multiple valve replacements, catheter-based, that are being looked at. These are just some of them. I quickly show you this Edwards Evoke one. We've done a large number of tricuspids on this. It has some anchors that kind of pull up the leaflets. It then has a bit of a sealing skirt around it, and then it has this frame around it above the atrium, so it kind of sandwiches this valve in. And the reason we need to do that compared to Tavr is that Tavr, as I said, anchors to the calcium. There is typically not much calcium in a mitral valve when it's leaking. It's more related to the leaflets being pulled apart, and therefore it has to anchor from kind of the top and the bottom to get a secure device placed that'll be there for a long time. I'm going to spend just a few minutes on tricuspid valve disease. As I said, this connects the top and bottom of the heart on the right side, where the blood comes in from your legs and the upper body, goes into the right atrium, through the tricuspid valve, out into the lungs, gets oxygen, comes back to the left side. So one of the things we've noticed is there's a large number of patients, almost 2.5 million cases a year, total cases, and about 250,000 cases a year are new. But look at this, the amount of surgery that is done for all of this is only 50,000 cases for mitral, and only 10,000 are done for tricuspid. So there's 217,000 new cases a year, but only 10,000 new surgeries done a year. And that's why many people have called this the forgotten valve. And people used to think, well, it was no big deal. Well, first of all, why isn't surgery done? And the reason is that morbidity and mortality is pretty high in this population. Many people have waited a long time to get treated because there weren't great treatments short of surgery. And so they've developed liver problems or kidney problems. And so surgery, you add that insult to them, and not many are done. So if you look at people 5 years after they've been diagnosed with severe tricuspid regurgitation, less than 20% actually receive surgical treatment. But same thing, and you're going to be experts now in looking at these afterwards, looking at a survival curve. Now we're looking at severe tricuspid regurgitation, and we're following people out. Let's pick the 2-year time frame, come straight up, and we look at 50%. Looks a lot like aortic stenosis. So once again, people were thinking, well, tricuspid valve, not a problem. But what we're finding as you watch and look at the data is actually this survival curves are not very good. I mean, half the people with severe tricuspid regurgitation will not survive out to 2 years. And even people with moderate tricuspid regurgitation have almost a 30% 2-year mortality rate. So we recognize here, once again, the only medications we use are water pills right now. That's the only that's even approved for use. And we know the surgical is not typically done. So we've been working on some things, and this is kind of when we get a clinical trial completed, we publish it in a journal. Obviously, then goes the FDA. This is one we just published about a month and a half ago in one of our major journals, looking at a new technology called Cardioband. I'm going to show you what that is. It was done in a number of centers throughout the US, many of the leading centers. This was really what we call early feasibility. So these are the first 30 patients essentially ever been treated with this device in the world are being included in this trial. And this is a kind of a busy slide, but I think kind of a really cool slide. This is us coming in from the right atrium, going across the valve. And what we do here is we put down a series of anchors, or they're nitinol anchors that we come around the entire circumference of the valve. As you can see here, here's the circumference looking at it. This is all an echo view of it. And now we put down, in this case, usually about 17 anchors. And once we have those anchors together, we can contract it down. So we kind of focus on that one here. Let me get back to that again. Let's get back to that slide one more time. Okay. So just kind of watch this one right here. You'll see how we contract it down. So those leaflets that were far apart, we've kind of pulled them together. We're looking at this under an angiogram. This is what these anchors look like under the angiogram. This is it here after we've implanted them all. And then you can see we do a contraction. So you notice this one compared to this one, you can see the band has been pulled together. And then I won't go through the echo too much, but this improved our leaking. And this was a study, once again, that we published in a recent journal article. What we know thus far with this technology is very limited. So I've taken you from one where we know a lot, we're now many years out, to we know some with the mitral, and now we know less with the tricuspid. So we're just getting started with these clinical trials. This was the early feasibility. What we could show is that we could improve that annulus. We could take it from 45 down to 39, so we could shrink it. What we also could show here is that using that NYHA class, how many patients actually felt better. You can see here that many patients felt better. And we do measure these things by p-values. There was a significant p-value for improvement in symptoms. And we could take the amount of tricuspid regurgitation. We have grades called torrential, massive, severe, moderate, mild. So we want to move the reds and the orange to the greens and the yellows here. And you can see that we did that in the majority of patients. Almost half were in mild to moderate. And if you took this group in here too, we're up to about 70-something% that had an improvement in the tricuspid regurgitation. Same thing here, looking at valve repair. This was looking at another clipping device called a clasp. This is a very similar, as I showed you before. It has these kind of paddles on it, and then we place these clasps down on the leaflets, and then we close this up just like I showed you that Mitral Clip. So it's a similar design, not quite the same. It has a spacer in the middle, but it functionally is trying to do what we call an edge-to-edge repair. In other words, pull the leaflets together. This was the very first data, again, early feasibility. You can see that 89% of patients were improved in their functional class, up to class 1 or 2, which is mild symptoms or less. The severity of the leaking valves, once again, moved over. Most of the patients moved to mild or moderate type of regurgitation. And their TR grade was also improved in a quantitative method as well. This is actually the very first US compassionate use case that I did almost 2 years ago. This is a patient that really had no options, was very sick. This is showing the leaking, all this blue that's coming back into the right atrium. We were able to put in a clip, 2 clips actually on this patient. And you can see the amount of leaking is almost minimal. You notice there's almost no, the dark blue is normal flow. It's this kind of really light aqua blue is what you don't want, and that red, yellow stuff. Those are the things you don't want to see. That's the meaning of leaking. You can see there's just a little fleck of red and yellow here. And the blue, the dark blue is kind of a normal flow across the valve. And same thing in this view here. So very first patient ever done. You know, she had really limited options and actually felt a lot better a few years out from the procedure. We talked a little bit about replacement. This is an exciting area for tricuspid valve disease. This is kind of looking straight down on this valve. This is the valve. We're kind of looking straight down the middle of it here. And this is looking at how it looks after replacement. This is looking on the echocardiogram. The echocardiograms become our guiding method for doing these types of procedures. So finally, last slide, and then we'll leave some room for discussion here. Tricuspid, remember I showed you on mitral, we save lives, aortic, we save lives. We don't know yet on tricuspid. We know we can make people feel better, but do we actually save lives? We don't know. What we do have is some data right here from a registry done in Europe, basically taking patients that receive treatment, so TTVI, meaning they got transcatheter treatment, and the blue line meaning people that just continue to receive medical therapy. And what we see here is that there looks to be a signal out at a year that we're improving survivability. Because this is not a prospective, randomized, multi-center trial, the data aren't collected as rigorously as far as how do people feel in the beginning, how they feel at follow-up, how well were the echoes controlled. All these things haven't been as well controlled. And that's why we need the clinical trials done as they're being done right now in the US with that clip, with the clasp, with the valve replacement, with the band, to find out how do those things do as far as survivability. And then another important endpoint we look at is survival free from heart failure. So not only did they live, but did they also not have symptoms of shortness of breath? And here again in this signal, we see that the group that was treated seemed to be doing better, 80% free of death or heart failure versus the non-treated group is only around 60%. But these data, we would not, as a clinician and a cardiologist, I would not accept this to say to a patient, this is something we should definitely use on you. We want more rigorous data to show that. But what we can use is this as a signal to say the safety looks high on it. We've been seeing that. And there is some signal that it may improve heart failure and survivability. But the clinical trials, what we're going to use to really understand that so that we can get approval and apply it to hopefully hundreds of thousands and millions of patients over the next 10 to 20 years. So with that, I'll pause and I'm happy to answer questions that I may have either confused or didn't cover. Thank you.
Thank you so much. That was great information and so many amazing innovations that are happening. I love to watch it. Jody, would you like to ask the questions?
Sure, we have a lot of questions actually. They're good ones too. First of all, Robin asked, is there a database for clinical trials existing or upcoming so heart failure patients can participate to help practitioners via their participation?
Yeah, that's a great question. The US government does have on their clinicaltrials.gov website all of the randomized and non-randomized trials that are ongoing in the US. So you can type in whatever you want, heart failure, valvular heart disease, coronary disease, and see the list that are approved by the FDA. So that's kind of the entree for, I think, that'll give you the overall what's out there. Now, those can be a little difficult to read through. That website is useful. It's not the easiest, and it's not the most friendly. I would tell you that the internet, though, in general, is a great resource of data for that. There's a website, heartvalvesurgery.org, that we've worked with Adam Pick. He's very good at putting out all the trials that are out there in one place. So that's a good location. I think you can go to many of the leading centers and at least look on their website. I mean, think about the top 10 centers. I won't sit here and name who I think of the top 10 or 20 or whatever, or some that may be local to you. Look on their websites. A lot of centers use that as a way of informing their patients. So there's various ways of getting at this. The industry also does it. I mean, so if you wanted to look at who are the leaders in the cardiovascular space and structural heart disease or valvular, it's Edwards, Medtronic, Abbott. Those are the ones doing it. They have some word there. So there's not one perfect repository, but there are ways of getting at that. The web is probably your best method to get educated on it.
Yeah, and so that answers another question of someone, how do we find out about trials if our doctors aren't aware of them? So definitely, but and you can always talk to your treating physician, obviously as well. Someone's asked, can you withdraw from a trial if you are not happy with the treatment you have been randomized to?
Yeah, so absolutely. That is, everybody has the right to do whatever they want. Just because you've signed a consent in a trial doesn't mean you are obligated to participate in that trial. And occasionally patients will remove themselves from a trial, opt out of a trial once they've consented and been randomized. I will say that I try to talk to patients about that ahead of time, because in order to have the integrity of a trial and accurate results, if people drop out after they've decided they were in but didn't get the treatment they wanted, it does impact our ability to understand whether treatment A is better than, equal to, or worse than treatment B, because we do analyze the data based on, was the patient consented and what group did they get put in versus the group that got by A versus B. So it really impacts the ability to get the correct answer from a study. If a large number of people did that, you'd never be able to show a difference of better, worse, or the same if people pulled out. So I try to have a discussion about that, and I think it's important for the patient to say, yes, I'm okay with whatever way this goes, and I would like to stay in it. But by no means I want to make sure it's clear that it's not an obligation, that you can't remove yourself from a trial. I don't know.
Okay, thank you.
Question.
Yes, I think that's good for people to know, and it is important, to have that data. So thank you for that answer. Sharon is a two times breast cancer survivor, and she has cardiomyopathy as a result of chemotherapy drugs, and she's taking spironolactone, bistolic, and varsartan, and wonders, and has a pacemaker, and is wondering how she finds out. And this is the broader question is, how do you find out if you're a candidate for a clinical trial? But I think she's talking about one of the trials you were mentioning.
Yeah, again, we receive referrals, sometimes self-referred from a patient. If they found us on a website or whatever, referral from their primary care doctor or referral from their cardiologist. And then depending on the trial, for example, with valvular disease, the echo is kind of the initial entree to understand what the pathology is in the heart, and then what treatments then might be available. So we usually work by collecting your records, and then work by obtaining any imaging that's been done already, and then decide whether there is an opportunity here to treat the patient, conduct a clinic visit, and then from there go from there. I mean, the nice part about telehealth, which has exploded recently, is that we can do remote visits with the imaging in front of us, and at least have an initial discussion as to, is there something we could potentially do here, and if we do, what are the next steps? And then it becomes the consent process and additional imaging that would be necessary.
Okay, thank you. We have two different questions, somewhat related, on outcomes of trials. One person participated in a trial and never found out about the outcomes. And, I know that people can participate in trials and feel frustrated of not hearing about the outcomes as well. And then one person would like to know, can you find out about, can you look at partner data and find out about the data and how it applies to specific populations like women, diabetics, etc.?
Those are both great questions, actually. First of all, the data are typically, especially anything registered on the US.gov website, FDA.gov website, those will publish, those data generally get published in the medical journals. I will say, as a whole, they don't necessarily, when the data come out, they're not necessarily provided back to the patient, not through any reason to, not be transparent. It's just not generally provided back to the patient unless you're back for follow-up and we have discussions with you. But the publications for any trial that you participate in will be also, once again, searchable, because they are presented at national meetings usually, especially the ones we're talking about here, and then presented in journals like New England Journal of Medicine, JAMA, American College of Cardiology. These are kind of like the typical ones we go for these types of studies. On the partner trial, it's actually been very well analyzed because they are such important trials in aortic valve disease. And all of those subpopulations have been analyzed. Women, elderly, poor heart muscle function, diabetics, multi-valve disease. There's literally over 100 publications looking at different subgroups. One thing as investigators, we always caution some of these subgroup analysis because, well, the trials weren't specifically designed, for example, to evaluate male versus female. So it becomes a kind of an after analysis, or what we call secondary analysis. And I think they provide important information, but we have to be a little careful in drawing full conclusions from a trial that doesn't specifically call out that characteristic prior to the trial. But the answer is yes, you can find specific subgroup data. Once again, if you type in partner trial and type in a subgroup, I mean, you'll find a host of publications. Most of them are free to look at. There's a few that do require medical access, but most of the big ones are pretty easy to get to. And then there's usually a lot of press releases around them as well that you can look at as well. It's a little easier to understand. Some of the other websites like WebMD, as I talked about the other one, heartvalvesurgery.org, some of those have some more information that's easier for a lay audience to read.
That's a lot of there's a lot of good data out there. It is hard to look at it all and read it all and understand it all. So thank you for that. One of our people who asked a question wants to know what valve Mick Jagger got, because he or she would like the same one.
Mick got a Sapien 3 valve. I didn't do Mick's Tavr, but I do know it was done, and Mick's been very, very open about his valve replacement, and also there's nothing here that's any violation of anything. So.
There's I think what is it?
I wish he lived in Chicago. Would have.
Well, he seems to be doing pretty well, so that's a good indication of that valve. Someone wants to know, and this is some of the various questions, do the trials measure what's important from a patient perspective in terms of quality of life versus only tracking the statistical survival data? It's a really good question.
Yeah, and I did kind of go through those a little quickly because I was trying to get through everything, but we do track quality of life measures, and that's actually been something in the last 5 to 10 years, which has been much more emphasized than just survivability. I always joke about, like, why do we have to have just survivability? Isn't it okay for patients to feel better? If you have a meniscal tear in your knee, you're not doing that because you're going to live longer, getting it repaired. You're doing that because you're going to feel better. So we have objective measures. One was that KCCQ, which is Kansas City Quality of Life Measurement. That one is used for subjective questions that patients are given to say, we give a baseline questionnaire and then ones that follow up to see how they feel. Another thing we use is that 6-minute walk, because that's an idea of how fast or how well you can walk in 6 minutes. And so that is another objective measure that we use. And then we have what's called NYHA, which is saying, can you walk a flight of stairs? Can you walk a half mile? Can you walk a mile? Can you just get out of bed to chair before you get short of breath? So those are the 3 most commonly used ones. I actually just finishing a study that we're doing here at Northwestern was using Fitbit on our patients before and after Tavr and looking at total steps each day. We found some interesting findings that we're in the process of publishing. But I think we more and more recognize that we want to get objective measures of well-being and not just survivability. And once again, if you search partner, actually we have a nice publication we did on that, looking specifically at quality of life measurements that were done and with the partner trial. And that was a whole manuscript dedicated to quality of life rather than just survival.
Another question is, I think this question is a feeling of a lot of people overall in terms of clinical trials. The last sentence is, I don't want to be a guinea pig. This is asking specifically about, I heard the mitral valve is more complicated to fix. What are the leading trials underway for companies with a good track record in this area?
Yeah.
And how would you know that in general?
I do think you should start with discussion with your primary care and your cardiologist and all to start beginning a discussion like that. The mitral valve and tricuspid valve are more challenging because they don't have that calcification that you can just blow up a balloon and put a valve in there and everything's going to be great. And there are multiple pathologies that cause that. I didn't even get into that today, but aortic stenosis is pretty much one. It's all vanilla. It's all calcified valve disease. Mitral can be from annular dilation. It can be from long-standing atrial fibrillation. It can be from abnormal leaflets. It can be from torn cords below. So there's a whole host of reasons why a mitral valve and a tricuspid valve can fail. And that's why you see this kind of toolbox that I showed you that were being developed with annular repair, with leaflet repairs, with valve replacement that we need to think about. The leading companies in these fields that have really done the most work have been, Edwards has really been absolutely at the forefront. Medtronic has some innovative products as well, and Abbott. So those have been the ones. Abbott's the maker of the Mitral Clip. Edwards has that Pascal Clip. Similar, not exactly the same, but technologies that both do edge-to-edge repair. All of the companies are working on valve replacements right now. Edwards probably has the lead on that right now, but you know, things right. And I've used a number of them and been very happy with the results, but I would tell you that it's very early on, too early to make, you know, firm conclusions on anything with that. So I think the important thing is marrying the company with the experts that really are dedicating their time to this. So it's the aortic valve, because it's easier, you can do a smaller number a year and be pretty good at it. Not, you know, I think if you do more, you're better. That's just my opinion, and I don't think it's a bias. I think it's a fairly valid opinion. But there's only, you know, a select group of centers throughout the country that do a lot of mitral and tricuspid disease. And I would tell people, if they're looking for catheter-based therapies for that they should be looking toward those centers that are really, that's what they wake up and do all day long. You know, they're not doing 10 other things. That's what that group of physicians, nurses, cath lab personnel, operating room personnel, anesthesiologists, radiologists, because it's a whole team. It's not just the one person that goes in and does a case. It's the CCU team. So everyone's recovering the patient, treating the patient. So look for centers that are very much involved with these technologies. They'll have more options for you, and that generally have more expertise in this area.
Would you include pulmonic transcatheter valve replacement in that group as well?
100%, because that's another one that's just not done a lot, and we didn't even talk about it today because it's in the adult world. It's fairly uncommon. Not a lot of centers are doing much of that. Many of the pulmonic work are also done with centers that are partnered with a good pediatric hospital because it tends to be more of a pediatric disease. And so I think that's another thing to look at, is if you're looking at that type of disease and some other types of structural disease, which are more congenital problems, to make sure that the center is very closely partnered with a pediatric hospital. Because there's even cases where, you know, we'll help them or they'll come help us. We happen to be attached to Lurie Children's Hospital. So we work very closely together on, if we have a pulmonic case where we just don't do a lot of those, we'll often work together on those. And I think that's in not just doing the case, but also deciding what to do for that case. Because it may be surgery, it may be catheter based. And so this is another one where we have a heart team discussion.
So on that same note with the children's hospitals, there's a question about, do you take results from adult trials and generalize them to children, or do you need to do pediatric trials separately? And also, what kind of diversity is required for trials? Is there a need for a diverse representation of patients?
Those are both excellent questions. So most of these trials, that at least all of the ones I've shown you today, were all adult-based randomized trials. There's a number of reasons why there aren't as many pediatric trials, not the least of which is most of these trials are patients that are already Medicare eligible. Medicare is required by law to be able to pay for a clinical trial if a patient's enrolled and gets a device in that trial. If a patient is less than 65, not on Medicare, then it's up to the private insurances to approve these treatments. And those always require special authorization to be done. So that is an impairment in getting patients enrolled into a trial. So much of the pediatric literature is based on what we call registries, meaning a number of patients were treated with X or Y problem. And now also because the prevalence of heart disease in the pediatric population is much less than the adult, it's difficult to even design a randomized trial where you get hundreds of patients on one side and hundreds of patients in another. And then there's the whole ethics about, taking someone that's a pediatric patient and randomizing in one or the other. So they tend to be registries, and many of them are utilizing adult devices in pediatric patients to try to treat some of these valvular problems. To answer the question about diversity, I think this is a really important question. And when we look at equity and inclusion, we recognize that many of the trials have a paucity of diversity to them as far as underrepresented minorities. And there's a number of reasons for that, not the least of which I think that often that population is not as well versed and informed, I would say, by their medical professionals on what options there may be available to them. I also think that clinical trials sometimes are more difficult to have underrepresented minorities agree to a randomized trial. I think sometimes there's a hesitancy there, and it does require a conversation. I think it's an important conversation. And I do see as we move forward in the last few years that there's more of an emphasis on increasing our enrollment of diversity in these trials. And I think the time is soon when we'll have a mandate of a certain percent, because I think you can't always generalize. We've had pretty good enrollment, I think, in many of these valve trials, particularly in the aortic valve, was actually pretty well represented by males, females, and underrepresented minorities. And I think we just have to keep working on this, informing the public well, and trying to recruit patients that may benefit, that don't have other options and may not be quite as aware of it by some of the ways that we discussed awareness of clinical trials. I think programs like this can help with that too. So I congratulate you in putting these forward and getting them out on the web, because the more we can inform the public, the more likely we are to be able to touch patients that may not have been aware.
Andrea, do we have time for another or no?
I think one more quick question and then we'll have to wrap it up.
Okay, one person asked, why are the deaths from all causes reported? Doesn't that cloud the effect of the Tavr procedure since it includes non-heart related deaths?
Yeah, that's an excellent question. We generally report both. I showed you all-cause mortality, but generally we report cardiovascular mortality and all-cause. The reason we use all-cause as the primary endpoint is because we can't always tell if someone dies what exactly the cause was. Obviously, we don't have autopsies in many patients. There can be a sudden death. There can be other things that cause them to die. And we have a hard time sorting out cardiovascular from non-cardiovascular. And because to remove any bias that we might have, we just go for all-cause. But recognizing, particularly as you get into these highest risk groups that have a lot of other problems, they can have bleeding problems, they can have kidney problems, they can have liver problems, they can develop cancer, they can develop other things. They just get lumped into all cause. The idea of a randomized trial is you would think you'd have an equal number in both groups. So if the trial is actually working, we're really sorting out the cardiovascular that way. But we do report it, but I think most people focus on the all cause just because we're not we're not perfect of sorting that out. We'll probably never be perfect at sorting that out. And we don't want to inject some bias into the study.
Great, thank you so much. And Dr. Davidson, thank you so much for your time. This hour was very amazing, and we got lots of great information. The next webinar in the series is next Thursday, June 10th, and it is at 3 o'clock, not 4, on Eastern Standard Time. And that webinar is going to do a deeper dive into what is involved in a clinical trial. So if you were thinking about participating, what would be involved in that trial? So join us next Thursday at 3 o'clock. And again, Dr. Davidson, thank you so much for spending the hour with us. We really appreciate it.
My pleasure. Thank you for inviting me.